Good afternoon. I'm Rebecca Driscoll, and I have the pleasure of serving alongside our team at Cancer Commons, which is a patient advocacy group serving advanced cancer patients. And today we are honored and privileged to be presenting with Travera, which is an exciting topic to learn about some therapy guidance for patients dealing with cancer. As it was stated, the new technology for rapid cancer therapy and guidance. And joining us today from the Cancer Commons team is one of our scientists, PhD, Kaumudi Bhawe.
Hi, everyone. I'm delighted to be here as the scientific representative for Cancer Commons and to welcome Travera today.
Thank you, Kaumudi. And then from the Travera side, we have presenting Cliff Reid, PhD. He's the founder and CEO of Travera.
Thank you, Rebecca. Happy to be here, and also like to introduce Dennis Watson, who is our Vice President of Business Development. For any of you who upon hearing this webinar, want to learn more, you'll be reaching out directly to Dennis, and he'll join us in the Q&A session at the end of the session.
Great. So just a few housekeeping items. At the bottom of your screen to ask questions, there is a Q&A button. You can submit a question anytime during the webinar. At the end, there will be a presentation of about 25 minutes, and you can present questions during that time. And the last 15 to 20 minutes of our call, and if it goes over a little bit, you can present that and we will answer the questions at the end. So feel free to do that, and we will take the questions in the order in which we received. So with that, I will turn it over to Cliff Reid to give us all of the information that they've got going on at Travera.
Great, thank you, Rebecca. So I'm going to do three things today in a space of say 20 or 25 minutes, and then we'll have the remaining time for questions. First, I'm going to describe for you sort of the problem that we are solving. Second, I'll give you a technical description of the solution that we offer. And I'll try not to drag you through too much math and physics, but it's a quite unusual and unique approach to cancer therapy guidance. And then I'll talk about the specifics of the program that we're doing with Cancer Commons. And for all of you who are interested, how to reach out to us and sort of take the next step of having your cancer cells tested. So here's the problem that we're setting out to solve. And I'm saying we're trying to, get the right treatment to the right patient at the right time. What I put up on the screen here is the NCCN evidence block for late stage breast cancer. So this is recurrent, unresectable, or stage 4 breast cancer. And what you see here is, a first line and a second line treatment. It shows it's trastuzumab, that's Herceptin, plus a couple of chemos, plus a couple of other drugs. And then you get to this third line treatment section. And there's a kind of a very, kind of unfortunate description here. And you see it in that yellow box. It says third line and beyond, optimal sequence is not known. And what's presented here is 12 different therapies in 8 different combinations. And the guidelines are, we don't know which of these are better or worse for the patient. The problem with this in particular is that this isn't just true for breast cancer. This page equivalently exists in all 61 of the NCCN cancers. The non-small cell lung cancer page is in some sense even worse because it presents 27 combinations of 13 different cancer drugs, and it lists them alphabetically. So that's really not cancer guidance. So that's what we're trying to do, is kind of address this problem, in particular for late stage cancer patients, of what should be their next therapy. Now, DNA sequencing was supposed to solve this, right? I was actually the founding CEO of Complete Genomics, one of the very early DNA sequencing companies. And in 2005, we set out to do a GLIVEC, for every cancer. GLIVEC is a genomic medicine for CML, for chronic myeloid leukemia, and it's curative. It's a stunningly good drug. And our mission was a GLIVEC for every cancer. Well, it didn't work out that way. It turned out finding targeted therapies for particular DNA mutations has been incredibly difficult. There are a lot of therapies, but there are a lot of duplicate therapies. And it turns out across all cancer patients in the United States, these targeted therapies benefit only about 10% of those patients. So cancer therapy guidance doesn't come from DNA, it comes from the NCCN guidelines, and only about 40% of the treatments administered under the NCCN guidelines actually help the patient. What we're trying to do is get to personalized medicine, but what we have is still population medicine. Even if you have a mutation, like an EGFR mutation, everybody with an EGFR mutation does not respond to osimertinib the same way. So we aren't yet to the stage of personalized medicine, and that's where we need to go. We need to get to the point where every patient is an N of 1, not part of a large population. And this problem is getting worse because of new drugs coming on market. I mean, there are 7 EGFR inhibitors. Osimertinib is frontline, but what about the other 6? There's no guidance for how to use them if a patient fails osimertinib. So we have this large and growing unmet need to match every cancer patient to the precise treatment that will work for them. Now, clinical trials are often kind of held out as, you know, an option for a late-stage patient who's running out of options under the NCCN guidelines. But the numbers there aren't good either. You know, fewer than 5% of adults actually get into a trial. About a quarter of them actually benefit from the trial. So you're benefiting less than 1% of the patients with cancer. And it's even worse in the rare diseases. So, you know, you're an N of 1, you deserve more individualized insight. That's the problem that we're solving. Let me tell you a little how we're doing this. I promise not too much math and physics, but you have to get a sense of it to know why it's so completely different than what's come before it. So our test is based on a new invention. It's a new measurement tool that was invented at MIT. And this measurement tool can do one particular thing. It can weigh a single cell, and it can weigh it with extraordinary accuracy. And give you 60 seconds of physics of how it works. What you see there in the picture on the fingertip is the device itself. It's a MEMS chip. It's manufactured in a semiconductor fab. And it's a fluidics device where we can pump fluids through it, and specifically pump fluids with single cells through this chip. What's inside the chip is what's called an oscillating cantilever. That's a really fancy word for a diving board. So there's a little diving board. You can see an artist's drawing of it there. And inside that diving board is a fluidics channel, a 30-micron fluidics channel, which is about the size of a single cell. And what we can do is pump single cells through this fluidics channel, and pretty quickly, about 500 cells a minute. And we take advantage of a very simple law of physics, and that is the resonant frequency of an oscillating cantilever is directly proportional to its mass. Measuring mass is hard. Measuring resonant frequency, that's easy. We can make frequency measurements using simple electrical circuits to an accuracy of about 10 parts in a billion. This gives us an extraordinarily accurate measurement of the weight of a cell, about 100 times better than anything anyone's been able to do before. And to turn it into more physical terms, we can detect the size change of a cell, the diameter of a cell, of about 5 nanometers. Okay, if you think about the wavelength of visible light, you remember that? 400 to 700 nanometers. This is a 5 nanometer change, about 100th of the wavelength of light. So it's incredibly small. This measurement tool has never existed before, and now we can weigh single cells. So what do you do with that? Well, MIT and Dana-Farber have been collaborating to do a lot of great things with it, but one of the things they collaborated on was to ask and answer the question, can we use this to measure which cancer drugs are going to be effective for which cancer patients? Okay, that is, can we take live cancer cells out of the patient, apply drug to them, and use the weight change of the drug to predict if the drug is going to be effective for the patient? And the last 10 years of research have turned out to say, yes, we can. It actually works. So, and it's based on kind of a somewhat simple observation that dead cancer cells weigh about half of what live cancer cells weigh, okay? But there's a terrible timing problem here. That is, when you take live cancer cells out of a patient, and particularly in the solid tumors, blood cancers are a little different, but in the solid tumors, you take live cancer cells out of the patient, they're dead in 2 days, okay? But if you apply drug to them, the drug is going to kill them in about a week, okay? But if you know that the dead cells weigh half of what the live cells weigh, you can ask and answer the question, hey, if their cells are going to weigh half in a week, might they lose a small amount of their weight, 3 or 4 or 5% in the first day while they're still alive, before we have to pump them full of drugs and cytokines and growth factors and other things, and you grow them into cell lines, grow them into organoids, which radically changes the behavior of the cells. They no longer behave like the patient. Can we make this measurement so quickly that the cells are still effectively behaving like they behave in the patient? That's what this measurement tool enables us to do. Okay? So here's kind of what we actually do, physically do. And I'll do this example for a single drug. We run panels of 20 drugs. But for a single drug, we would take 10,000 live cancer cells, split them in half, 5,000 in each well of a plate. Add drug to one well of the plate. Don't add drug to the other well of the plate. Keep all of the other conditions identical. And then wait for 24 hours, essentially incubate those cells with that drug. And at the end of the 24-hour period, weigh each individual cell in each well. Don't weigh the wells. This is not a bulk measurement. It's a single cell weight measurement. So we weigh each individual cell in each well. And what you see in the bottom right of the slide is these two sort of bell-shaped curves. These are the weight distributions of the cells. And the cells are in all different stages of the cell cycle. So they span quite a range of weights. But what you see is the drugged cells have a slightly different weight distribution than the undrugged cells. So if those two weight distributions were the same, that means the drug doesn't do anything. The drug isn't working. But if those two weight distributions are different by an amount that was too small ever to measure prior to this new measurement tool from MIT, if they're different by a little bit, then we know that those cells are on their way to dying, in a week, even though we're making this measurement in a day. That's the breakthrough technology here. We don't have to grow the cells. We don't have to keep them alive for a week in order to test them. Okay? So kind of an obvious question that raises is, how well does it work? How well does it predict patient behavior? And the answer is quite well. So here what we have is, we're a very young company. We don't have a lot of clinical validation data yet. I'll show you, we have 49 patients that we've run all the way through our system. And the data represented in this slide is gathered by us getting a live sample of the patient's cancer cells. We run a panel of drugs against those cells. The oncologist gives the patient one of the drugs that we've tested. Months later, we get the results of the clinical outcome of the patient. We then compare that actual clinical outcome to our prediction, made months prior to the outcome happening. And if the two line up, if they're the same, we say we got it right. And if they don't, we say we got it wrong. And of the 49 patients that we've tested so far, we've gotten 41 of them right. So it's a better than 80% predictive accuracy. And you can see what we've done here is the myeloma patients, AML patients, and then solid tumor patients, about a third, a third, a third. And we make both positive and negative predictions. A positive prediction means, we predict the drug will work. And when the oncologist gives the patient the drug, it actually does work. Negative prediction, we predict this drug isn't going to work. Oncologist goes ahead and gives it to the patient and it doesn't work. And you see we've got, both of those in there. We're very thin right now on solid tumor data. We've only done 17 patients, got 15 of them right. So I want to be very clear that this is a very early stage test. And so we're building up our clinical validation evidence. We're learning which cancers we're better at and which cancers we're worse at. But the amazing thing about this test is because it's a cell level test, it's not a molecular test. All the carcinomas, breast, lung, colon, pancreas, they all behave pretty much the same to us. They might be molecularly very different and different genomics to them, but at a cell level, they all behave about the same. So we can do a lot of different cancers with the same test. Here they are. So we are CLIA certified right now for 39 of the 61 NCCN cancers. Very specifically, we have not yet validated the lymphomas, the sarcomas, the glioblastomas, or any of the pediatric cancers. But we are CLIA certified for all of the carcinomas, so that's 85% of all cancers right there, multiple myeloma, and we're just finishing AML as we speak. And what you see in the bottom of the slide is the drugs that we've tested. So we've done timing and dosing studies of every one of these drugs, so we know what dosage to use in our lab, so that we're getting a kind of the most accurate readout we can of whether or not the cells are going to respond to the drug. And you see, a large number of targeted inhibitors, the classic cytotoxics, the chemotherapies, AML and MM drugs, and then on the right side there, you see the immunotherapies. This is new. We're now running the four major PD-1, PD-L1 checkpoint inhibitors in our lab. We're running these in patients and returning the results. We simply don't have enough predictive data yet to know if our measurement predicts the behaviors or the responses of cancer patients to these checkpoint inhibitors, but we're gathering that data as we speak. And this is what our CLIA lab kind of looks like in a workflow. So you kind of know what's going to go on kind of in our lab if you end up participating in our test. So what we do is we need to collect a live cell biopsy from the patient. And today, The live cell biopsies we're collecting are malignant fluids. So these are pleural effusions of the chest cavity, and the ascites of the abdominal cavity. We are just about to launch our tests for solid tissue samples. Those will be out in Q1, and we actually hope they'll be out in January. But for the purpose of this program, we're only doing malignant fluids right now. If you want to do a tissue sample, we can get your name, you'll send it to Dennis, and we'll notify you when we're ready for you. But the idea in the current workflow of our lab is we take a malignant fluid. You would take the malignant fluid and put it into our standard shipping kit, into little vials in there. You close up the kit, press the button to start the temperature control, and take it into FedEx. It would ship overnight to our lab. We would take this vials out of the kit the next morning. We would plate them out. We would add the panel of 20 drugs to certain wells, and then use the others as controls. Incubate overnight, and then run every one of those cells through our measurement tool, through the SMR, and weigh each and every cell from each well. We also have sort of a fancy, it's called a CNN classifier, where we take a picture of every cell or every cell-like object going into the measurement tool. And sometimes these cells they're broken up, they're really debris. Sometimes they're clustered together. And by taking pictures of them and using an AI-based classification system, we can reduce the data set down just to the good, whole, intact cancer cells. We weigh the cells, get the weight distributions of drug versus no drug, compare those weight distributions, write it out into a nice report, and send it back to the oncologist. There are two wonderful clinical characteristics of this test. The first is that it's really fast. If you do a biopsy on Monday, the oncologist has the results on Wednesday. It's a 2-day turnaround test, 1 day to ship and 1 day to incubate. And secondly, we only need about 5,000 cancer cells per drug tested. So that's 100,000 cancer cells to run a panel of 20 drugs, which is a relatively low number of cancer drugs. Okay. And here I'll show you what the results report looks like. Sorry for the very small font, just to give you an idea, though, of what it looks like. Page 1 of our results report is the list of 20 drugs, in the panel, ordered by how well the cells respond to those drugs. And they're scored on a scale of 0 to 100. A score of 100 means these cells strongly respond to this drug, and a score of 0 means these cells don't respond to this drug at all. This is an actual patient report. We ran this patient about 2 weeks ago. It's de-identified. This is a non-small cell lung cancer patient. And on page 2, what we see is a little more data. The vertical axis remains the score, 0 to 100. Horizontal axis is the actual mass change of the cells. This is a physical measurement. We're measuring physically how much do these cells respond to these drugs. And in this case, I will say, we get this result. It's not always what you'd expect. I mean, if you look at the drugs that scored 100, it's a whole bunch of the targeted inhibitors that are on label for non-small cell lung cancer. But one of the targeted inhibitors, trametinib, which is a wonderful drug, scores 0. This is not something the oncologist would know ahead of time. Similarly, if you look at the chemotherapies, which patients tend to respond to reasonably well, here they are down at the very bottom. There's cisplatin, carboplatin, paclitaxel, docetaxel, etoposide, the classic chemos that a lot of patients respond to. This patient responds to none of those. And yet, way up at the top, here's doxorubicin, a chemo. So you see results from this test that represent the actual behavior of likely heterogeneous cancer cells to a set of cancer drugs, and the results are often not exactly what you expect to see. Okay, so I'm going to change gears now and talk about the program itself and how you can participate in it. So we are, thrilled to be partnered with Cancer Commons, and one of the key things we're doing is we're providing this test to the Cancer Commons community free of charge. Partly because we're very early stage, we don't have a huge amount of clinical validation data, hence we are not CMS reimbursed yet. We're working on all of those things, but patients who are willing to work with us and run the test help us build up our clinical validation data, and hopefully we help them find the next effective therapy. We do have one ask for you if you participate in this early access program for free, and that is give us your clinical outcomes. So you will be getting an online survey. It's fully HIPAA compliant, takes about 10 minutes to complete, can be done by the patient or a caregiver or other representative. And that link will show up sort of in a few months after your test, where you've had time for your oncologist to know if you responded or didn't respond to the test. So who qualifies? Well, first of all, you need to be eligible for a new therapy, because cancer therapy guidance doesn't help if you can't change therapies. Very importantly, as I mentioned before, this is this is for malignant fluids only at this time, okay? The solid tissue samples will be available in early 2023. And at the end, you can send us a note and tell us that you want us to contact you when that time comes. Also, this is a CLIA test. It has to be ordered by a physician, and it could be a medical doctor, mid-level provider, and also as a nuisance factor. We're very new. We've applied for CLIA certification everywhere, and it's been granted in 48 states, and it's pending right now in New York, Maryland, and also in Washington, D.C. So here's the summary. This is a free test through Cancer Commons. Your physician must write the order for this test. Because it's a malignant fluid test, you need to collect 3 50 milliliter vials of fluid from a pleural effusion or an ascites. The idea is to collect these during normal fluid drains. We're not reimbursed, so you can't write an order for a new collection or a new biopsy. This needs to be a clinically indicated collection, and tissue is coming. You place the sample in our shipping kit, and we'll send you a shipping kit. All you do is put the sample in the kit, press the button that starts the temperature control, seal the kit, and take it to Federal Express for overnight delivery. Has to go out the same day. These cells die so quickly that if it isn't shipped to us the same day, the test probably won't work. We will run 20 therapies and the 4 immunotherapies also against your lab cancer cells. And then the following day, we'll deliver a report back to your physician, giving the list of all of the drugs that we tested in the order of their likely effectiveness. Then you and your physician can use that information to select the next therapy. And at some point, you'll be asked to fill out a survey about which therapy you ended up selecting and how you responded. So here are the next steps. You need to tell us if you're interested. So on the next slide, I'll have email addresses, and you can reach out to us or reach out to Cancer Commons, whichever you prefer. And just reaching out to us doesn't mean you're committed to doing it. We understand you may just have a lot of questions, and we're delighted to answer them. So if you're interested, you'll contact Dennis Watson, our VP of Business Development. And Dennis is very comfortable working with you and with your physician, meaning you can either tell your physician you're interested, or you can tell us. And then we can reach out and contact your physician and begin to educate them, because chances are your physician hasn't heard of us. We're very, very new in this marketplace. So with that, I'm going to move to the question session here and kind of open it up to the questions that I think you've been sending in the chat. And Dennis and I will speak for Travera. And if you have questions for Cancer Commons, we'll go directly to them.
Thank you so much, Cliff, for this wonderful presentation. And thank you, Dennis. And now I'd like to sort of start off with some of the questions we've been receiving in the background as the talk was happening. So one of the questions that has really come in, this is first of all very interesting, very, very new and groundbreaking. Who pays for the sample collection? For example, is this included as part of the test, or do patients have to ask their insurance to pay separately for this?
It is not reimbursed, and it's not included by us. So what we are strongly recommending is that you collect these live cancer cells as part of a clinically indicated biopsy that's already been ordered by your physician. So if you have a pleural effusion and it's going to be tapped, largely for reasons of comfort, then when that tap happens, we ask you just get 150 mils of that tap and send it to us. We're steering patients away from doing specific biopsies for us because there's no reimbursement and we can't help pay for that. Perfect.
So this is good to know. So if already there is, it's happening, then there shouldn't typically be an additional cost that's attached to it. Okay.
And Kaumudi, just to add to that a little bit, some patients that do have a pleural effusion or ascites of the abdomen often have a catheter placed where they maybe even drain at home, or they drain with home health or something like that. And we can even work with you in those situations, but it should be something that's done as part of normal care rather than a specific order related to this assay.
Perfect. Dennis, thank you. This ties in with another related question we have here, is that, for example, if a patient has already a malignant pleural effusion with a Pleurex drain, can they take the sample themselves and send it out to you, or what happens? How's the logistics for that?
So yes and no. They can take the sample. However, because it is a CLIA approved test, we do still need a physician's order to be able to run the analysis and run the assay and report on it. So, you know, I could work with the patient directly, even on how to communicate with their doctor about this. I do that often, and I'm more than happy to provide some guidance there. But it does require the physician's order. Once the order is in place, the method of collection is less relevant to us, as long as it's done appropriately and properly.
Perfect. Okay, thank you.
Yeah.
So these are good questions around actual sample prep and requirements. One of the patients has asked, for example, can someone be actively participating in a clinical trial and still participate in this?
That's up to the clinical trial, but our answer is yes. And just even more generalized, we don't have any restrictions on prior treatments or current treatment or anything like that. We'd like to know if the patient is on a current treatment, because we're going to see that in the drugs that we test. But if they're participating in a clinical trial and there's no prohibition by that clinical trial for doing a test like ours, then it's perfectly fine with us.
I had a related question actually for both of you, since we see patients that come to us early after diagnosis, we see patients late stage, you know, the whole spectrum here at Cancer Commons. So is there a typical time frame within which you want this done, meaning right, immediately after diagnosis or after multiple therapies? Does it matter? Is there,
you know, from our testing perspective, it doesn't matter at all, but I will share some sort of practical perspective on that. And that is early stage cancer patients, newly diagnosed cancer patients, we find are often working in a very, you know, fruitful and positive manner with their oncologists. And they're given a lot of very precise therapy guidance from the NCCN guidelines. Mm-hmm. And at that point, they feel like they're in good hands. What we found is that as a patient progresses, and particularly the patients that get to stage 4, and they start beginning to understand what I showed at the beginning of the presentation, that the therapy guidance provided by the NCCN guidelines becomes, you know, limited or non-existent. And then what they're really doing is that their oncologists are running, you know, doing kind of trial and error experiments over a large number of therapies that they don't really know which ones to pick. That's where the value of our test can be very high. But having said that, there is nothing preventing a newly diagnosed patient from using this test if their oncologist is, you know, supportive and willing to write the order. So.
Mm-hmm. That's good to know. Okay, along those lines, is there any plan to sort of combine this with the liquid biopsy tests that are available, or how do you see that?
Yeah, so unfortunately that doesn't work because, you know, liquid biopsy, which is really a blood draw, typically something like a 10 ml blood draw, doesn't have enough cancer cells for us to test. I mentioned that our test, to do a full panel of 20 drugs, we require about 100,000 cancer cells. Well, the number of cancer cells, you know, not in the blood cancers, but for a solid tumor patient, typical number of cancer cells in a blood draw, in a 10 ml blood draw of peripheral blood, is about 10 cells per milliliter. So a 10 ml blood draw would get us about 100 cancer cells, where we need 100,000 cancer cells. So I regret to say we cannot run this test from a liquid biopsy test. We must have a malignant fluid or a tissue sample.
That makes a lot of sense. I think this would speak to one of the questions that did come in. This particular question was that, what happens if someone comes in without any known cancer? For example, could a healthy person test? Could this be a screening test, for example?
No, it couldn't, because once again, we need 100,000 live cancer cells. And so if we were screening against some body of cells, none of these cancer drugs are going to work. You know, these are, you know, for regular cells, they won't respond at all. And somewhat related point though is, what if a patient doesn't know what kind of cancer they have? What drugs do we run? Well, there's an NCCN guideline cancer called the occult cancer, which is a cancer of unknown primary, which is simply a case where you may have a metastasized patient and they have no idea what the tissue of origin really is. And in that case, you know, we run a panel, sort of a balanced panel of chemotherapies and targeted inhibitors, plus the 4 checkpoint inhibitors. So that's a situation where effectively there's no guidance at all, and we can really provide that cancer patient and their oncologist some important guidance.
And also, Kaumudi, is as part of our purification process, when we initiate our test, we do an analysis on the cancer cells present in the sample. And so if there are no cancer cells there, then it would actually fail our quality control, and it wouldn't even proceed.
Okay, that's good. Thank you.
Yeah.
So one of the patients asks, I mean, this is related, can you send blood? So the answer is probably.
Well, we do test multiple myeloma.
Okay.
And so the bone marrow sample we are CLIA certified for, and we're just about to release AML, and that'll be from blood, but those are two very special cases. For solid tumors, we won't accept blood.
There's a lot of questions that have come in around whether you work off of liquid samples only or solid tissue as well. Can you speak to that a little bit, the different sample types that
Again, the sample types that we support are the malignant fluid, so pleural effusions of the chest, ascites of the abdomen. And then we're about to release the product for all tissue samples. And those tissue samples would include fine needle aspirates, core needle biopsies, and surgical resections. Again, we find most of our patients are very late stage, so we see very few surgical resections, but we do see fine needle and core needle aspirates. And we find that, for example, in non-small cell lung cancer, relapsed patients will often get biopsied a second time to check the genetics of the relapse, to see if there's some new mutation that has a new targeted therapy. And in that case, we would love to jump in at that moment and say, hey, send some of that sample to us so we can test a whole series of drugs against it.
Right, perfect. Thank you, Cliff. So this ties in with another question, is that have you sort of done any cross-testing of your results with the recommendations that are generated by NGS testing companies, for example?
Yeah, yes, we have. And in particular, the way that's mostly done, again, we've only had 49 patients. So there's not a lot of drugs, not a lot of patients that we can test, but we've tested a huge number of cell lines. And we will, get a cell line that has a particular mutation in it, such as an EGFR mutation, and we'll then run, osimertinib against that cell line and show, using old technology, just using flow cytometry technology that takes a week, that's all working well. And then we run our mass measurements side by side, to figure out, are we getting the same results as the old lab equipment, and we line up on those to those measurements over 95% of the time. Our measurements are give exactly the same results that a targeted inhibitor would give on a cell line that's known to be sensitive.
Excellent. Okay. So for patients, do you have? I mean, I see that you presented that there's some pre-specified panels that you use, but on a per patient basis, is there a way to sort of customize this, or do you have a standard test always, standard set always? Do you shift that around?
Yeah, we actually offer both. We have the 39 standard panels that are on our website that you can go look at, and we also have a bespoke product, a completely custom product that will allow a cancer patient and their oncologist to pick 20 drugs of their choice. They may be 20 off-label inhibitors. However, for this early access program that we're offering through Cancer Commons and offering for free, in that case, we're limited to just the standard panels. It would be too overwhelming to our laboratory to try to offer custom panels for each patient. So we'll just pick, the most appropriate of our standard panel for the Cancer Commons patients.
Very understandable. Related to that, there's about close to 300 FDA approved drugs, but you have focused on a subset of that, 100, and then you have panels of 20. Mm-hmm. How have you gone about choosing this?
Yeah, that's a great question. We just did all the popular ones. And we're just, next year at this time, I think the number will be 200, and 2 years from now, I think the number will be 300. So I think we're going to go do all of the cancer drugs, but quite a few of those 300 drugs are a bit obsolete. They're not used very often anymore. So we've done the most popular ones. I will say there's one category of drug, though, that we haven't released yet as part of our CLIA certified tests, and that's the antibodies. We do have an assay in our R&D lab that is working. It's too complicated for us to run in a CLIA environment. So one of our key jobs in 2023 is to simplify. that assay so we can launch it as part of our CLIA environment. So for example, multiple myeloma patients, frontline therapy now is 𝗡𝗲𝗄, which is an antibody. Well, DARE is running beautifully in our research lab, but I regret we're not allowed to use that in our CLIA certified lab because we haven't done that validation, that simplification. But other than the antibodies, we've run just about everything, and it'll just take time to work our way all the way through that list of 300 drugs.
Thank you. So, okay, this is a little bit of a tricky one now. Can the results of this test be used with insurance companies to get their approval for certain treatments in certain specific cases where you need that sort of extra?
The answer is we don't know. It's never been tried. Because of the extremely limited clinical validation data, we would not expect an insurer to be very favorable to this test. I think they'll say, we'll come back when you run hundreds or thousands of patients. And that's part of why we're running this early access program, is to generate exactly that clinical validation data and to make this test available, early to patients who really are out of therapy guidance from the NCCN guidelines. So I think eventually it will be very helpful, and we're going to go for CMS, reimbursement just once we have enough data to submit for that. But we don't have enough data to do that now. So I'm afraid this is unlikely to be enough evidence for an insurance company to reimburse for it.
Okay. One of our attendees has asked, once you get to solid tumor testing, will the preferred method of obtaining the sample, is it going to be FNA or core needle biopsy, just to clarify?
Well, our preferred method is more cells are better. But we have nothing to say about that, about what the patient is going through. So we will take either. And if you send us a patient sample, say from an FNA, and it turns out it doesn't have 100,000 cancer cells in it, it has 25,000 cancer cells, we'll run 5 drugs. So it kind of degrades gracefully. We just want as many live cancer cells as we can get, so we can run all of the drugs that we want to run and the 4 checkpoint inhibitors, from the T cells that are in the sample. It's, and that we love the malignant fluid as sample types because we often get 1 million cancer cells out of malignant fluid, and we get the T cells that enable us to run the checkpoint inhibitor. So more cells is better, but we'll take any sample size, any sample type that you have access to, and we'll do the best we can with what you send us.
With the malignant fluid samples, does it vary from cancer to cancer, the number of cells that you harvest? And
yes, it does, and that's in the literature. I'll give you a couple of data points to kind of describe it. For pleural effusions, chest cavity liquids of non-small cell lung cancer, over 80% of those are full of cancer cells. Over 70% of breast cancer patients with pleural effusions are full of cancer cells. If you go to ascites and go to pancreatic cancer, and we have a lot of interest from pancreatic cancer patients because the disease has so little therapy guidance coming out of the NCCN guidelines. If you have an ascites for pancreatic cancer, only 50% of those have cancer cells in them at all. And then if you go to an ascites for a breast cancer patient, only 20% of those have cancer cells in them at all. So it varies widely by cancer and by biopsy type. And Dennis has all these statistics at his fingertips, and will work directly with the patient to either say, hey, go ahead and send us that, we're pretty confident, or ask you, have you ever had a histology test? Do you know sometime in the past that there were or weren't cancer cells in your malignant fluid? Because it turns out patients are amazingly stable in whether or not they have cancer cells in their fluids. If you had a histology test 6 months ago or a year ago, it still probably is correct for your current fluid. So if it said, yeah, that fluid a year ago was full of cancer cells, we would say, send it to us regardless of the population statistics. That's a personalized thing for you, and we'd be delighted to take that sample.
Perfect. Yeah, I was going to follow up with that, because that's very important. I mean, that is a question. As we all know, tumors evolve, cancers evolve over time in a given patient. There's heterogeneity, but you're saying that you've seen that the cancer cells harvested from the fluids sort of stays stable over time. That's what you're experiencing.
Yeah, if the mechanism of generating that fluid is cancer, it will be kind of full of cancer cells. If the mechanism of generating that fluid is sort of an organ blockage that isn't directly, the tumor itself, then there's probably no cancer cells in there, and there never will be. So.
Okay, here's one on a slightly different topic. Do your panels, you kind of covered this, but just do your panels only include FDA approved drugs?
Yes, they do, because but you, as you can tell, we're not putting the drug in the patient, so there's no prohibition, and we do research in our same lab, running these same tests where we're using preclinical drugs. But for our CLIA certified laboratory, we have only CLIA certified the FDA approved drugs. Yeah.
So if a patient is interested in the research aspect of it, they just contact you and on a one-on-one basis. They
contact us directly, and we can run non-FDA drugs against samples, but not return them as part of our CLIA report.
I see. Right. Of course.
Yeah.
Of course. So if a particular drug has already failed to work for a patient, and it's on your panel, do you still include that, or can that be replaced by something else now?
What we can do is change the order of the drugs that we're running. That turns out to be very easy in our lab. So, and it just happened to us last week that a patient had just failed a month ago, one of the top 5 drugs in our panel, and we just took that drug and moved it to number 20. Now, having said that, it's still awfully interesting to run drugs that patients have failed before. If they've just failed it immediately, it's often kind of confirmatory to say, yeah, sure enough, your cells aren't responding to this drug. But if a patient failed a drug, a year ago or 2 years ago, as you said, these cancers are heterogeneous and they're moving around. And it may be that a patient essentially has a population of cells that was knocked down a year or 2 years ago that has regrown and has become resensitized to a particular drug. So we have some flexibility there about what order we run the drugs in, but we often encourage patients to say, no, even if you failed the drug, let's go ahead and run it again and see what happens.
All right. So you did mention the cancers you're currently able to work on. Do you have plans to address glioblastoma anytime in the future? Or?
We've only avoided glioblastoma because it's so hard to get the cells, and the way to get cells from glios is through surgical resections, and that just hasn't been something that the community has told us about, that they were willing to do a lot of that. So there's nothing technically preventing us from doing it. It's just not a place where we expect to be able to get live cancer cells with enough frequency to offer the test.
Understand. We have one of our listeners has come in with a comment rather than a question, but this is an interesting comment. So the person says that all patients with my kind of cancer begin with carboplatin paclitaxel, which involves months of misery before finding out, with the recurrence that the chemo didn't work.
Yeah.
And so they say that I hope someday every patient with carcinosarcoma will be able to take this test before treatment. So any thoughts both of you want to share?
We agree. I mean, I always say, think about the positive and negative testing. If a drug works in our hands, if we find your cancer cells are sensitive to our particular drug, that's not an absolute guarantee it's going to work for the patient because of drug delivery, right? Because our drug delivery in our lab is perfect. We're taking known quantities of active molecules and putting them directly on the surfaces of cells for a known amount of time and making a terrific measurement of it. Our drug delivery is perfect. When you put a drug into a patient through an IV, the drug delivery isn't perfect. And maybe that drug that works in the lab never gets to the tumor or gets to it in such a concentration it does no good at all. So those things we're going to miss on occasion. It's just endemic. We'll never have a perfect test because of drug delivery. But on the negative testing side, if we take your live cancer cells, put them in a lab, hit them with a drug like a cisplatin or a carboplatin or paclitaxel, and it does nothing, chances that that drug is going to work in you are really slim because our drug delivery is perfect, right? So the negative prediction that where you shouldn't subject a patient to a toxic drug because we have a lab test that shows it's not going to work, we think is one of the great values that we can offer.
And in fact, even though the numbers are small right now, as you guys saw, if you recall from the earlier slide that Cliff showed about the negative prediction versus the positive prediction, our negative prediction in solid tumors right now is above 90%. It's very good. And so I agree with him wholeheartedly that that's going to be a really important value that this test will be able to bring, maybe even to early stage patients.
I sort of have a follow-up question out of interest now on this, is that with the negative testing, how I guess again, this falls along the lines of numbers, right? To convince the treating physicians, because they're the first ones who have the responsibility of deciding for patient, what could be the next line. So how do you actually make a convincing case for a negative result? I guess that must be.
Well, at this stage, we don't make the case strongly at all, because we say we all have to be very realistic about how much clinical evidence there is that validates this test.
Exactly.
It's very small. So all of the patient testing that I showed in that slide of 49, it was not interventional based on our test. It was observational. So this is, but you know, when we get a patient sample sent to us, we know the stage of the disease and we know the type of cancer it is. And that already limits it down to a pretty small set of drugs. So we run 20 and we're almost guaranteed to hit the drug that the physician is then going to prescribe. So we can get real kind of prospective data, even though it's not an interventional study. So I would say we need to generate a lot more data before we want any physician to sort of change, particularly for an early stage patient where there is, you know, good population-based guidance as far as that goes, but we need to do a lot of validation before we want those decisions to be made by oncologists for early stage patients.
Thank you for clarifying that. Thank you. Yeah. So
I'm sorry, real quick, if you don't mind me sort of intervening. I know we're sort of at the top of our scheduled time. Cliff and I are more than happy to stick around and answer more of these questions, but I thought it might be great to sort of put a bow on just a couple of little details. I noticed there's a lot of questions in the Q&A about the type of, you know, the type of sample, et cetera. And so, you know, in respect to this partnership and you know, this work that we're doing with you guys, anyone right now that's listening to this that is interested in getting this testing done, and you do have either a pleural effusion or an ascites, please email me directly. My email address is on the screen. It's dwatson@travera.com, and I will work with you and hold your hand as much as is needed to help you communicate with your physician, to provide you resources, communication, support, et cetera, to make sure we get that done. And we'll work with you really closely through that process to make it as simple as possible. Regarding the tissue-based test, that, as Cliff said, is very close. We've finished all of our validations. We're just really optimizing it now. And so, you know, we're expecting that as early as, you know, late January, but certainly in the first quarter. And so if anyone is interested in that, please also just send me an email and just let me know that you're interested in that, because what I can do is add you to sort of a wait list. That way, when that becomes available, I'll be more than happy to follow up and you know, circle back up with you and let you know that that's available. In both cases, just like with the malignant fluid, we do recommend it being done as part of a normal procedure, because since this isn't a reimbursed test, the procedure won't be reimbursed. But as Cliff noted earlier, that often happens. You know, you're getting restaged, you're getting new genetic analysis run. The docs are already in there. It's not a huge deal for them to take you know, an extra core, an extra this, an extra that. And so that's what we try to sort of piggyback on with those. There was a question on here about bone mets. We cannot do bone metastases. It does have to be soft tissue. So I wanted to answer that one as well. But I just wanted to sort of clarify some of those points. I know there will be a follow-up email going out, I believe, but feel free to email me if you're interested in any of this. And we can certainly go back to the remaining questions with the time we have remaining.
Absolutely. Thank you, Dennis. And everyone can see the email on the slide here in front of you. So that's something to take note of. Yeah, so we only have like 5 to 7 minutes left before we're at the top of the hour, or a little more than that, but I'll insert a few of these questions here. So is there any particular cancer type or drug that you have tested that best predicts a favorable clinical response?
Best predicts a favorable clinical response.
Is that too hard, too early to answer?
No, yeah, we don't, again, not enough data, but you know, I don't think we're going to find, you know, anything that is a panacea. You know, that's a drug that,
as we all know,
for everyone. I think, you know, again, most of our patients are heterogeneous, and finding, you know, 1 or 2 or 3 drugs that moves the needle is real victory. But you know that, you probably are familiar with the statistics. Even the genomically indicated drugs, the targeted drugs that have a mutation, they typically only work in often 40 or 50% of the patients that get them. I mean, you have an EGFR mutation, you get an EGFR drug, and it works half of the time. We're hoping to improve on that. We're hoping to be able to say, well, that mutation is part of all of the complexity going on inside that cancer and being addressed by that cancer drug. But if you think about what a live cell is, sort of the ultimate integrator of all the different kinds of information that make up response, because it's taking into account not just the genomics, both the known genomics and the unknown genomics. We still don't know everything about genomics, but it's taking into account the proteomics and the epigenomics and the metagenomics and all of the different forces operating on cells with drugs. So our goal and intent is to have a test that dramatically improves this sort of 40% success rate that averaged across all drugs by personalizing the test, by not saying, hey, this is a population study. No, this is how your particular cells are responding to this particular drug at this moment in time. That's the design of our test.
And the ability that you're able to get in early and do this in a quick turnaround time, I think that's also.
We've had a lot of positive feedback about the 2-day turn. Often oncologists are really under the gun to make a decision for a patient, and most tests kind of operate in the 2 weeks kind of turnaround time. So 2 days is terrific, and our turnaround time won't get any longer because the cells will be dead. We are technologically and biologically forced to make this test work in a 2-day turnaround time.
Yeah.
And Cliff, I think that brings us to I'll go ahead. I'm sorry, Kaumudi.
No. Go ahead, Dennis. And then I'm just going to add in one last question, and then I just want to sort of.
Yeah, no, I was kind of doing the same. I think it brings us to another important point that I noticed was a question in the Q&A, which is how do the oncologists handle this test, and what do they do with it, and do they use it? And what we find in our experience is that when they receive these results, they use them to guide therapy in many cases in our experience. That said, Cliff just made the point that we're unique in the way we do our measurement, and if you were to take this to your oncologist, what you might find is that they have a general belief that this type of drug testing just doesn't work, because we've been trying this in cancer since the 50s. The 1950s. But that said, we've always tried it by growing cells after they were collected from the patient, and in some manner or another. And that growth process, that intervention, changes the cells. It changes the cancer. As you guys probably all have learned in your own personal journeys, cancer is essentially a mutant, and when you intervene with it, it mutates further. And so what we're doing is unique because we're really the only test that's ever existed that is testing real drugs against the cells directly extracted from your body, not some later iteration of them. And that's what makes this special and unique. And if you face that kind of headwind with your oncologist and you just need some guidance as to, hey, I understand this is different, but I don't understand why, that's the kind of thing we're more than happy to help you sort of navigate.
Perfect. I have one pressing question from one of the audience members that I'm going to ask here, and then I think the time is running up, so we will be wrapping up after this one. So and there will be, I mean, you can always reach us by email. You can reach Dennis or Cliff, and you can also reach out to us at Cancer Commons here if you have follow-up questions on this video or on how we at Cancer Commons help patients through these kind of different testing technologies or their treatment plans. We work on a one-on-one basis with patients throughout their cancer journey. So you can contact us as well. The question is, does your technology enable the study of drug efficacy from a combination of drugs used simultaneously? Or?
Yes, it does in 2 ways. One that's available and one that we're going to release next year. The one that's available is we can actually run drug combos. And if you anybody with multiple myeloma, if you go and look at our multiple myeloma panel, we run KRD and RVD, and we run the standard drug triples in our lab. We also run the popular chemo combos, so FOLFOX, FOLFIRINOX, FOLFIRI. And it, one of the things that's very interesting for us to run, and again, early stage, is to run FOLFIRINOX, which includes, oxaliplatin and irinotecan, and then run those drugs as monotherapies too. And if FOLFIRINOX works and irinotecan works and oxaliplatin doesn't do anything, drop it, and just run the drugs that work and avoid the toxicity associated with the drugs that don't work. So those are the kinds of things we can do in combo therapies today, literally combining the drugs in our laboratory. The second thing that we can do, it's a research topic we're working on, and it's just one of the most exciting things we're doing, but not yet ready for product release. And that is, we can show that under the appropriate circumstances, which really involves heterogeneous cancers, that these drug responses add, meaning the weight change adds. It's an additive thing. And because it's a real physical, meaningful quantity, the weight is changing. And if one drug changes the weight by 3% and another drug changes the weight by 3%, and what you really need is a 5% weight change to call that drug effective, the combo will change the weight by 6%. And so we are experimenting and exploring and running all sorts of validation tests right now before we're willing to assert that this is clinically valid and that we can actually predict the behavior of drug combinations based on testing the monotherapies and then calculating the effect of the combo. So watch this space. We're not ready to go with that, but I think it's the single most exciting project we're working on.
Well, thank you so much, both Cliff and Dennis, both of you, for coming out here and giving this very informative talk to all of our listeners. And with that, I just wanted to say that everyone is welcome to reach out to us with questions to either Cliff or Dennis or to myself or any of us at Cancer Commons. Thanks everyone for joining us today. And with that, I'll call it a wrap.
Great. Thank you for having us. Bye-bye.